Good morning, good afternoon, everyone. My name is Tiantian. So today, I'm very honored to be here to share my work as part of the microTAStic seminar series. On this special day, I'm excited to share our latest work on the fusion of liquid-liquid phase separation and microfluidics. So droplet microfluidics has been around for quite some time, and it is a transformative platform that allows us to create and control tiny droplets with remarkable precision. So in a typical setup, we need two immiscible liquids. So these two immiscible liquids are brought together, and one forms the droplet, the dispersed phase, while the other acts as the surrounding fluid. So the strength of droplet microfluidics actually lies in its ability to control the flow while maintaining distinctive interface between immiscible liquids. Typically, water and oil stabilized by surfactants are paired to create thousands of uniform droplets per second. These droplets, each of them can serve as a container for discrete reactions. They can carry cell, reagents, or complex biochemical mixtures, revolutionizing high-throughput analysis by reducing sample volume and speed up experiments. So this is quite the crucial advantage for techniques such as single-cell analysis and digital PCR. And they can also function as soft templates. So by loading as a soft templates for material synthesis, for instance, by loading droplets with a polymer precursor, and later on triggering gelation through temperature or UV light, each droplet transforms into a microgel with a defined shape and size. This is quite useful for fabricating new materials. So in recent years, people actually find in living cells a different kind of droplet, not formed by water and oil, but formed by liquid-liquid phase separation. So people find that in cells, some proteins and RNAs, they cluster into dense membraneless droplets, such as P granules and stress granules. These droplets, they dynamically assemble and dissolve in response to changing cellular conditions such as pH and temperature, so allowing the cells to respond and reorganize its biochemical processes. And they can also concentrate specific molecules while excluding others. So these droplets actually provide an efficient means to compartmentalize reactions, and they therefore give the cell a capability to respond to signaling cues and demonstrating how nature used droplet-like structures to finally control when and where vital reactions occur. So inspired by these droplets we see in cells, we can form water-in-water droplets, employing synthetic liquid-liquid phase separation. So on the left, you can see that combining a polymer that carries negative charges with a polymer carries positive charges, they spontaneous lead to droplet formation. And the phase diagram in the middle shows that the droplet formation in a certain concentration and conditions of pH, temperature, and ionic strengths. And if the condition is right, the mixture will split into a dense droplet phase and a surrounding continuous phase. So here you can see in the microscopic graph that it's a clear demonstration that these are condensed droplet in the solution. So the most interesting thing about this water-in-water droplet, or we call it coordinate droplet, is their diffusive interface and their remarkable ability to selectively recruit certain molecules. So here so here you can see a test tube containing a coordinate base, and when we add CY5 labeled DNA into through the diluted phase, the DNA molecule becomes visibly enriched within the droplets, as shown in the red fluorescent image on the right. The fundamental reason behind this selective enrichment is that the droplet interior provides a unique chemical environment. So the molecule the molecules can partition into the droplet only if they can favorably interact with the environment, or they can favorably interact with the specific scaffolding material already in the coordinate droplet. In other words, these coordinate droplets are not inert droplets. Their soft dynamic interface allows certain molecules to enter while excluding others. So this tunable partitioning is very useful for for understanding membranous organelles, also for applications if you want to do target encapsulation, bioseparation, or controlled chemical reactions. So another distinctive feature of these coordinate droplets is their ability to assemble in response to various chemical cues. For example, here we show that at low pH, no droplets, but at higher pH, a dense droplet phase suddenly forms. And if you reverse the pH shift effectively, you turn off the droplet again. And these droplets, they can also transit from a liquid state to a gel-like state. So on the right, we see if we heat the droplet, it causes the droplet to melt. The heating causes the droplet to melt and fuse, while cooling induces gelation. Although we know that water oil droplet, they often show similar behavior, but here is a water-water system. So they can do reversible and tunable transitions from no droplet to droplet and to gel, make it very attractive platform to develop smart and bio-inspired materials. So these coordinate phase, they can also form multi-compartmental structure if we are using multiple coexisting aqueous phases. So these coexisting aqueous phases can be incompatible dense phases or arising from multi-phase liquid-liquid phase separation, which combines segregative liquid-liquid phase separation that's typically occur between natural polymers or and associative liquid-liquid phase separation that's typically driven by multivalent interactions. So here is a photo demonstrating 3 coexisting aqueous phases in a single container, and while the fluorescent image highlight that different molecules can enter specific compartments, and we envision that this multi-compartmental droplet structure can be very useful for multi-step reactions. And subsequently, by incorporating these 3 coexisting aqueous phases in a microfluidic setup, we can form uniform droplets. Here, aqueous phase A and B, they meet a dilute phase stream and resulting multi-compartmental coordinate droplets whose internal structure can be precisely tuned. So notably, coordinates, they typically exhibit strong shear thinning behavior, which helps the jet breakup and consistent drop formation. And so during the drop formation, interfacial tension is a crucial parameter guiding how these coordinate droplet at equilibrium organize internally. So we know that phase separated systems typically have very low interfacial tensions because standard techniques may lack the precision to detect these at very small values to obtain their quantitative value. We in the lab develop a very simple, we call it particle levitation technique, which we hang a suitable particle at the soft liquid-liquid interface. So by comparing the gravitational force pulling the particle downward with the interfacial tension holding it in place, we can accurately calculate, quantify these tensions. Here we can make it down to 10 to the minus 8 Newton per meter scale. So this information is vital because interfacial tension directly influence the internal organization of the droplet. So after forming droplets, we're focusing on how to stabilize phase separated droplets in aqueous environment. This is a challenge because traditional surfactant don't work for these water-water systems. So in living cells, we see a clever solution. Protein particles like those in P granules, they absorb onto the droplet surface and prevent infusion. So this surface decoration effectively give the membraneless organelle stability and functional control. So borrowing this strategy, we also introduce particles to the water-water interface. So the key requirement is that protein particles, they have to possess suitable surface property to anchor themselves at the aqueous-aqueous interface. If they bind too strongly to either phase, they will get excluded or engulfed instead. We find that protein nanofibrils, they effectively absorb to the droplet surface and forming a jammed shell and preventing fusion and leakage. In addition, interfacial mineralization of calcium carbonate can create a rigid particle-packed layer. So this is another strategy for stabilization. And these protein particles, they often have pH regulated partitioning, so which allows us to tune the stability of these droplets. So in the fluorescence image on the left, you see distinctive dyes labeled 2 aqueous phases, red and green, as well as the protein particles we use are kerosene nanoparticles, they label blue. So depending on the pH, these kerosene nanoparticles can co-localize with either phase A or phase B while absorb out the interface. So the key is that their partitioning coefficient will shift dramatically with pH, enabling us to actively control where the particles are relative to the protein's isoelectric point. We find that these kerosene nanoparticle decorated coercive droplets can be very stable in aqueous environments. Even after heating to 8 degrees C for 2 hours, the solutions undergoes a minimal change, and the bar chart here shows that droplet size stays nearly constant. So meanwhile, we compare different protein stabilizing agents in the literature by plotting their tolerance to pH, time, and the temperature. So the comparison reveals that these kerosene nanoparticles are very stable. They maintain their structure over a year now, in even under harsh conditions. So this is ideal for demanding applications that require a long shelf life of water-water emulsions. So aside from protein decoration, we also like to show that an unexpected way to stabilize water droplets without adding any extra stabilizers. We show here that microflows generated by evaporation can maintain separate cold shell water-in-water droplets. As you can see in the schematic, we place a sensor droplet on a oil-wetted substrate and simply let it evaporate. Over time, we observe a ring of cold shell phase-separated droplets forming at the edge of the original droplet, and remarkably, they remain distinct and stable with no additional particles. Typically, you need a stabilizing agent to prevent coalescence, yet here the evaporation-driven microflow effectively keeping these droplets from fusion and preserve their sizes. This shows the self-assembling route to achieve stable and hierarchic and complex droplet structures. Next, we investigate why these droplets, stabilized droplets, appear specifically at the liquid-liquid air contact line. So actually here, evaporation is the most intense at this boundary, driving convective capillary flows both up and down to replace lost fluid. And this flow creates a ring of stagnation points where niche flow is minimal. So in this zone, smaller satellite droplets remain painted. They remain painted and preventing themselves from fusion with each other. So essentially, these droplets are trapped in place by localized microflows. So after we get stabilized the droplets, next we show how coercivate droplets can act as miniaturized microreactor. Here we perform a reaction DNA amplification using lamp reactions. So here at in the top images, you see that lamp is run inside these droplets. They glow under fluorescence, indicating successful DNA amplification. Meanwhile, a control without lamp shows no fluorescent signal. We also compare reaction condition in two types of water in water droplets, one with aqueous two-phase systems, the other using coercivate. Here we like to point out that in the coercivate system, lamp reaction is actually depleted in the dilutive phase. In other words, the reaction only happens inside the droplet, whereas in ATPS, they occur in both phases. So crucially, each coercivate droplet functions as its own micro reactor. So that means the reaction in one droplet don't interfere with the other. So by concentrating, this is because coercivate droplet, they can effectively concentrate in DNA primers, and enzymes in these dense droplets, they host highly efficient reactions. And we also show quantitative power of using coercivate droplets for lamp-based DNA amplification. So here in this image, each bright green spot represents a droplet where amplification has occurred. You can see that the initial copy number increases, so that's the number of the fluorescence droplet. So next we show that we can actually using these droplets to perform multicellular like assemblies or to support inter-droplet reactions. So taking inspiration from the hang drop method using in cell sphere cultures, first we use microfluidics to produce lipid-coated coercivate droplets, and these droplets, they can they self-assemble, then we self-assemble them through capillary forces. And so because these surfaces are lipid coated, so neighboring droplets, they form interfacial bilayers. And these interfacial by carefully controlling both the number and type of the lipid coated droplets in each cluster, we can have this droplet clonings, and these lipid bilayers, they connect each of the droplet in the cloning into a larger biomimetic assemblies. The key role of this lipid bilayer is to regulate and control what goes in and out of each droplet, much like the cell membranes do. And this allows us to coordinate signal transmission and enzyme cascade reactions among these droplets. We can effectively replicating droplet to droplet and droplet to environment communications. And this approach can extend it to form hybrid cell and protocell spheroids as a new kind of biomaterials. Next I want to share a little bit on what we use this phase separated system for bioprinting. So typically when you extrude soft bioinks layer by layer, they cannot support their own weight. So especially when you do the upside down or overhanging structures, stiffer inks, you can do that, but that's not ideal for living cells. So a lot of people are using suspension bioprinting or embedded bioprinting. So we see that the phase separated system are very good for embedded bioprinting. So typically we use phase separated system and aqueous phase A as ink and aqueous phase B as matrix. So then we also introducing yield stress characteristic to the matrix phase, and this method is very effective. To both very advantage, both good at maintaining complex geometry and ensure the cell viability, because it allows you to because of the low surface tension, it allows you to use low viscosity inks. And using this strategy, we can actually form free-form all aqueous 3D structures, all in a water environment. And because the low viscosity inks are used, so that's very gentle on cells. If we place living cells in either the ink or the matrix, we can compartmentalize different cell types and nutrients with the same printing constructs. And building on this water strategy, we also can make the surface coercivates, so that we incorporate charged polymers such as positive and negative charged polymers into two aqueous phases. And due to the electrostatic interaction between these polymers, they can cross-link at the water-water interface, and the cross-linked structure can support not only stabilize the structure, they can also support the cell growth. And this interfacial coercivated membrane can form a interconnected perfusible microchannels, particularly valuable for tissue vascularization. So here in these images, you can see branching vascular structures that can incorporate living cells in it. As an example, we co-seed endothelial cells in the ink phase and smooth muscle cells in the matrix during jet extrusion. And then we make the interface coercivation. This interface coercivation actually function as scaffolds that support cell attachment and the culture. So after 3 days, we can see that the inner and outer surface of the hollow fiber membrane form a confluent layer of the endothelial cells. And these cells, they express correct biomarkers, and we test their barrier functions and confirming they effectively mimic native structures. So I think because these free-form printed networks are suspended in liquid medium rather than embedded in a gel, so it can be handled as independent tissue constructs. This is very useful for thick vascularized tissues and other biomedical applications. So to summarize, we use biological ingredients such as proteins, nucleic acids, and cells combined with multiple liquid phase separator system and microfluidic manipulation, and we can achieve organized compartmentalization and function. And by leveraging tailored coercivation, we can form multi-compartmental droplets, build microreactors for biochemical reactions, and engineer perfusible vascularized networks. So we envision this powerful synergy will enable the creation of more functional in vitro models and provide deeper insight into replication or more accurate replication of complex biological processes. So these are some of the papers we published. And so we are a small group in Shenzhen University, which is very close to Hong Kong. And we have 3 PIs in the group, and most of our students are master students. And we also work on other research topics such as biomimetic hydrogel robotics. And thank you for your attention.